Okay, children, let me continue the same lesson, breeding and biotechnology. This is the second unit. Okay, first unit we have seen what is plant breeding, what is animal husbandry, how to meet out the needs. Then who is father of green revolution, how Dr. Swaminathan made revolution in India, and what are the varieties, what's the staple food of India, wheat as well as, right, wheat for North Indian, rice for South Indian, and what are the nutrients that new varieties should be supplied to us, that is protein, oil and mineral. Then what are the methods to be followed to make a development in the plant production. First one is introduction. New variety from all over the country we are getting the new varieties, that is known as what? The new varieties of plants we are collecting, that is exotic species. From the exotic species you have to select the best one. So selection also, three kinds of selection: mass selection, pure line selection, clonal selection. What are the three kinds? Mass selection alone, how many there you see? Mass. Next one, pure line. Third one is clonal. You see how the selection differ from each other. First one you see, mass selection. Seeds of the best plants showing desired characters are collected from a mixed population. All are mixed together, no? You have to select which is the best. And we have to select that kind of plants. Mass means what? Group. And the group of plants, and then we have to select the best one. Okay, the collected seeds are allowed to raise. First generation we are sowing the seeds. From the seeds we got the flowers, again we get the seeds. That is second generation. Like that, generation to generation. Can you follow, children? Generation, we are sowing the seeds into the plant. They grow, they produce the seeds. They produce the flowers. Flowers shrink ஆகும்போது நமக்கு என்ன கிடைக்கும்? Seeds. That is first generation. Second, we are getting seeds now. That we are sowing again. That is second generation. Like that, we are making up 7 to 8 generation. So the seeds, the 8th generation what you got is, how many generation it comes over? 7 generation followed, and the 8th generation it will be very selective and supportive to the human being. So we have to select this kind of things. At the end, they will be multiple and distributed to the farmers for cultivation. Immediately we cannot get. So for example, it takes one year also. First generation, second generation, for example, every month they are producing. Like that, 8th generation, we collect the best seeds and give it to the farmers. Some of the common example for the mass selection are groundnut varieties, TMB2, AK10, then representation given saying multicolors of seeds are there. They are selected which is the best. Red in the first generation. The red seeds they are producing again. For the first generation they have taken 5 seeds. 3 plus 3, 6 red color you see the diagram. 6 seeds they are selected. That is the best seeds they follow. That is the first generation. Second generation the same, like 6 seeds. And the third generation, fourth generation, like that it will be multiple. The 6 seeds, from the 6 we got lot of seeds that can be given to the farmers for the cultivation. And the pure line selection, progeny from a single plant, single individual by the self-breeding. That is the same plant. For example, jasmine you have taken, jasmine to jasmine only. So though we are selecting only one. The mass selection we have taken many characters, but he has taken only one kind of plant, jasmine, that to what? White jasmine, for example. The white jasmine adhe da edukringa for generations. So this is pure line selection. So father and mother both are, that is male and female both are jasmine only. So there is our new organism also what? White jasmine only. Like that it can be. Pure line it is. Then clonal selection. So pure line is both genotypically as well as phenotypically same only. Phenotype I said already, externally, outside, outlook, and genotype internal. Both will be same. Then clonal selection. This is only one, no, pure line. This is not like that. By the vegetative method or asexual method, plants can produce lot of seeds. Okay, that can be collected. The clonal method, from the same plant, making up, going on, dividing them, we are getting the Xerox copy. From one copy, we are getting many Xerox copy, no? Can you differentiate the Xerox copy and the main copy? No. What all in the main copy should be as it is in the Xerox copy. The same, like, from one seed, we are producing many viable seeds together. It is known as what? Clonal selection. Clear? So next we have to say polyploid breeding. What are the breedings I said? Polyploidy, mutation, and then orally you say, no? Polyploid, mutation, and then what? Hybridization. We'll see what is polyploid, what is mutation, what is hybrid variety. Now we have to see what is polyploid. Polyploid breeding. Here we have seen already. We have got chromosome. How many pair of chromosome? 23 pair of chromosome in the human beings. The same like plants also got the chromosome. Okay, the chromosome we got two kinds of thing. One is somatic cell, other one is generative cell. Somatic cell is body cell, generative cell is reproductive cell. We have two complete set of chromosome, that is somatic cells, they are diploid. Okay, somatic cells, body cells. In all the cells, what should be there? Nucleus. This is diploid, that means two nucleus in everyone. And germinal cells, that is one only, that is haploid. Why this is haploid? Because both the parental characters are fused. So one cell, it should have minimum two nucleus only. So the somatic cell, there is no chance of fusing together. So they have two nucleus. Clear, children? But reproductive cell is not like that. There should be two characters should be mingled, male character and female character. So they are haploid in nature. By fusing male from one, female from one, altogether it will be diploid. Clear? This is called diploid. An organism having more than two nucleus, two sets of chromosome is known as polyploid. Polyploid is more than two. One means diploid, haploid, polyploid. That is more chromosomes. Somatic cell has diploid chromosome. Germinal cell has haploid chromosome. Other parts of the cell, they have more chromosome, that is known as polyploid. So sometimes because of the chromosomal divisions, they may be happen like that. So the things which helps to polyploid is known as physical agents. Physical agents. Then achievements of polyploid. By producing these kinds, what's the achievement? What are the uses? That is seedless watermelons. Because of the protection, that is what I say. Diploid, haploid and polyploid. What is the main functioning of polyploid? To bring the improved varieties of the fruits and as well as vegetables. For example, what are the benefits of the polyploid breeding? We get watermelons. Watermelon, you know what it is? That is seedless watermelon. The watermelon, we got lot of seeds. But in the polyploid breeding, all over watermelon, seedless. Seedless watermelons and bananas. Polyploid, seedless watermelon and bananas. You are familiar with the fruits. Clear? And next one, TB29, triploid variety of tea with larger shoots and drought tolerance. Next one, tea. We are getting the tea, triploid. Three flavor. Triploid variety of tea, that is TB29. It will be stand for long time. It is drought resistance as well as it produces more also. And triticale, this is hybrid variety of wheat. We are getting from that. It has higher dietary and fiber protein. So by consuming this wheat, we got dietary. Dietary means what? Fibrous fruit. Fibrous is good for the digestion. And Rapo brassica is allotrophied by cocaine treatment. Okay, these are the new type of plants. Next one, what is it? Mutation breeding. Next one is mutation breeding. Mutation, sudden changes. Mutation means what? Sudden changes, already explained this is. Mutation is defined as sudden heritable change. Heritable means what? Generation change in the nucleotide sequence, the DNA in the organism. Changes, sudden changes in the DNA. DNA, deoxyribonucleic acid, which is the genetic material. It is the process of genetic variation and created the changes in the organism. For example, we can see some people, they have the same 6 fingers. Some other extra growth they can see in the thing. This is because of sudden changes in the body. And we have seen eunuchs. That is, they are not considered as male as well as female. This is also mutation only, sudden changes only. No eunuchs, no. But and we used to say Ali, whatever it may be. They have the characters like us only when they are in the child, they got male characters or female characters. When the certain age come, when the teenage come, they have some changes in their body, the behavioral changes. Though they are male, they differ, they think like a female and dressing up themselves. This and that what? Behavioral changes. This is because of chromosome. This is mutation changes. That and all harmful. But we are cultivating by ourselves mutation breeding, which will be useful to the human beings. So, this organism which undergoes mutation is known as mutant. And the agents which are help to the mutation is known as mutagenic agents. Clear? Then what are the mutagenic agents? DNA mutation. What are the thing is there? One is physical. Physical mutagens which support and chemical mutagens. Which is supporting for the changes. Physical mutations, that is because of radiation. Physical means what? Externally. Because of radiations like X-rays, alpha, beta, gamma rays, UV rays, temperature, etc. Okay, these are the causes for the physical mutation. What are? X-ray, you know what it is? Alpha, beta, gamma ray. The radioactivity, it can release the rays, that is alpha rays, gamma rays and beta rays. UV rays that we are getting from sunlight, ultraviolet rays, temperature, everything. Because of that, we got the sudden changes, that is physical mutation. Extra X-rays, alpha, beta, gamma, this and all known as mutagens. And chemicals mutagens that induce mutagens because of the chemical appliances. For example, which are the thing making up mustard gas and nitrous acid. These are the thing which changes in the mutation. Achievements of mutation breeding. Because of achievements, what are the thing? We got many varieties. Savarabadi sonara, that is the wheat, that can be produced by gamma rays. Automata 2 rice, that is saline tolerance and pest resistance. Saline tolerance means what? Salt. Evlo uppu thannina the spoil agadu. That no pest can be affected it. Groundnuts with the thick shells. Thin shell illama shell in outer cover of the groundnuts. By opening the shell one, we can get the groundnut inside. The next one we have to say hybridization. Hybridization, I said already, 2 different organisms we are taking out. 2 different superior quality of organisms we are taking out. We are fusing together and from them we get a new organism, that is new offspring. This is known as what? Hybridization. That is 2 or more types of the plants for bringing their desired characters together into one progeny called hybrid. Progeny means young one. New plant or new organism, whatever you can see. That is different plant. For example, hibiscus, red color hibiscus and yellow color hibiscus. Red is one thing. Yellow color hibiscus is other one. Red color hibiscus, maybe the flower is very thick. Just will imagine. Red color hibiscus is very thick flower. Yellow color is disease resistant as well as production is more. Red color, attractive, help for the pollination, for example. This is the character. For that only we are selecting grade. We are selecting the characters. Why we are taking the hybrid red color? Because it is very attractive in color, helps in pollination. And yellow color, it produces more, it yields more, and it is disease resistant. எந்த ஒரு நோயும் வராமல் இருக்கும். ஸோ போத் we are fusing together by mixing of red and yellow DNAs and we are getting orange color hibiscus. This orange newly we are getting, that is known as progeny, and this new organism is known as hybrid. Clear, children? This is hybridization. Hybridization, we like that we are finding out meaning. That is, what are the things we are getting from the hybrid? Beet, rice. I'm simply telling this and not beet rice. Mainly we are making up for the satisfaction of the food sufficient of food satisfaction of human beings. We are doing all these methods. Okay. So the characters finally obtained. They got both the character. This is recessive character and dominant character. Dominant வெளியே வருது, recessive அடங்கி இருக்குது. Finally, dominant character will be released. Okay. Next we have to see animal breeding. So plant breeding, do you understand, children, what it is? So 3 methods of adopting new one. That is polyploid breeding, mutation breeding, and then what is it? Hybridization. Now we have to go for, so far what all you have seen only for the plants. Because what's the food of the human beings? Plants as well as animals. Now we have to see what are the modern techniques used in the animal breeding. So plant thing is known as breeding. Animal husbandry, we will see what are the things. Clear? We will go for the second part of the lesson. First part is over. First one we have seen what is breeding for the plants. Now we have to go for animals. Animal breeding. First we'll see the definition for breed. What is breed? A group of animals of common origin within species that has certain distinguished character that are not found in other members of the same species, like general appearance and other striking features. A new variety of thing. Breeding I know, because we are mating the parents, that is male and female. Mating means what? Fusion of the male and females, and we are producing different varieties. That is known as breeding. Male, female, new, that is known as what? Breeding. They produce the new organism, that is breeding. Then objectives of animal breeding. Then what are the aims? Already we have seen plants the same like animals. What are the aims of that? Aims of animal breeding. Improving the genotype of domesticated animal to increase their yield, to improve the desirable qualities, produce milk, egg, meat. What are the things we can get from the animals? Milk, egg, meat. That should be a superior quality. Breeding takes place between 2 ways. Animals of the same kind, animals of different kind. Same kind of breeding means that is same kind of animal, tiger to tiger, that is inbreeding. Tiger to some other animal, that is outbreeding. So breeding further we can divide them into 2. One is inbreeding, other one is outbreeding. Inbreeding with the same species, outbreeding different species. Okay. So what is inbreeding, you see? Inbreeding refers to mating closely related animals within the same breed. Same breed we are making up. For example, no example is given there. For about 4 to 6 generation. For example, elephant to elephant. We are getting elephant only. This is going on for so many generations together. Superior males and superior females are selected to give the better new organisms. Okay.